think I first met Edvard probably at poster sessions at the Society for Neuroscience meetings in the 1990s. But for sure, I remember meeting he and May-Britt Moser in 1995 in Oslo at Per Andersen's what he called Grand Slam event. Per called it that. He had 6 of his students finish up at approximately the same time, and I don't know how he managed that, but he did. And over a week, they all publicly defended their PhD theses. It's quite a formalized process in Norway, with not only internal thesis committee members there to critique the work, but Per invited 12 scientists from around the world to act as opponents, as they're called. And Edvard's opponents were Bruce McNaughton and Tim Bliss, and I was one of May-Britt's opponents along with David Amaral. So naturally, these 2 young scientists were spectacular, but each of the 6 students was given a particularly hard time as possible by the opponents, which is the opponent's job. But the reward in the end for the PhD candidates and the opponents was an amazing sleigh ride in the snow and a marvelous feast afterwards. May-Britt and Edvard spent some time in Edinburgh with Richard Morris and then in London with John O'Keefe, both during and after their PhD defense. And in John O'Keefe's lab, they learned the basics of single cell recordings. Although they'd planned a longer postdoctoral period, maybe even a stay in Tucson, an amazing opportunity arose for them for building independent laboratories in Trondheim at the Norwegian University of Science and Technology, or NTNU. There were, I have to say, mixed opinions on whether they should pass up this opportunity to do more postdoctoral work. Trondheim is a small city compared to Oslo and very far north in Norway, but Bruce and I encouraged them to go for it and just do it and to build something that was independent and absolutely their own. And looking back, it obviously was completely the right thing for them to do. So in 2001, Edvard, May-Britt, with their 2 daughters, Isabella and Eileen, came to Tucson for a sabbatical to learn parallel single unit recording methods that Bruce and I had implemented in the laboratory that the University of Arizona actually explicitly built for us for this purpose. And while Bruce and I had the ideas for how to achieve these kinds of recordings way back in 1981 when we were postdocs in John O'Keefe's lab, it wasn't until Tucson that all the component parts necessary for high density recordings came together in a functioning system. So around that time, the Norwegian Research Council inaugurated a new funding scheme where 13 centers of excellence were selected among all fields of science and technology and would be given extensive funding for 10 years. In December of 2002, Edvard and May-Britt were awarded one of these center grants that created the Center for the Biology of Memory in Trondheim. As part of this award, they included funding to support 7 international advisors who would come once or twice a year to help with experiments, help advise them and their students. And I must admit, I really enjoyed the yearly visits over that 10-year period. Typically, I would go in January, a month that other advisors would avoid because of the short days. But to me, it was a wonderful time. People were back fresh and keen to go after Christmas break, and the cross-country skiing is fantastic in January in Trondheim. So it was during this 10-year time period of the Center for the Biology of Memory that the Mosers and their students discovered grid cells, a fantastically exciting discovery in neuroscience. And of course, the Mosers were awarded the 2014 Nobel Prize in Physiology or Medicine for their discovery of grid cells in the entorhinal cortex, along with John O'Keefe, who discovered hippocampal place cells. I believe Edvard is going to review some of the history of this for you today, along with updating you on newer, exciting discoveries that continue to come from these NTNU laboratories in Trondheim. Okay, over to you, Edvard. You can share your screen.
Okay, so thank you very much for that nice introduction, Carol. And you're absolutely right. Had you not been in Oslo that evening in 1995, we would not have gone to Trondheim. Maybe I wouldn't have been at this conference now.
Yeah, thanks.
All right. let me now see if I can share the screen. So can you now see the screen?
Yes, great.
Okay, good. All right. So let me then start. I first want to start here because I really had looked forward to getting back to Tucson. There's so much that I learned there during a very dense period in, I think, 6 weeks in 2001, where Maj-Britt and I learned everything about large-scale neural recordings. And this just shows one of the fun memories that we also had outside the lab, walking back up in the Catalina Mountains. Is that the desert behind Carol's house? So I hope so much to experience that again, but not this time. I still hope it will be possible soon. So I was invited to speak at this conference, and the first thought that struck me was that I'm the wrong person, because I'm very hesitant to have any strong opinions about the neural basis of consciousness. However, I think we share an interest in understanding the highest levels of cognitive function. And I mean, there's nothing that maybe has been more interesting to humans through history than to understand the origins of the human mind, or as we might say today, the neural basis of the spectrum of intellectual or cognitive functions. So the approach I have taken to this is that I want to understand some of the simplest cognitive functions, and spatial orientation is among those. I think it's a useful place to start for anyone interested in in general algorithms underlying cognition, because there are so many elements now that have been described, parts or pieces of the space system in the brain, that I think everything is in place for mechanistic insights to take place. So I'll take you through our journey through the mechanisms of space, but and time. I will also come to time. But let me start out with a little bit of history, and I want to go all the way back to Tolman. So Edvard Tolman was a psychologist who worked at University of California, Berkeley in the 1930s, 40s, and 50s. And he made a major proposal that is totally fundamental to all the work that I will be describing to you now. So what he suggested was that not only our knowledge of space, but all our knowledge is structured around based on internal maps of the environment. Those are spatial maps, but they are maps that also include knowledge about things that happen to the places. So it's a way of organizing knowledge. And he based these claims on experiments where rats walked in mazes, and where he found that they take shortcuts, they make detours, and do not just repeat habits of behavior. So this was very, very controversial at that time, when behaviorism was at its peak. But it paved the way, and although there was no evidence for such maps for a long time, for several decades actually, it formed a fundament for the idea that there are maps of our environment in the brain. And this then was followed by a breakthrough in the early 1970s when John O'Keefe and Jonathan Dostrowski recorded from neurons in the hippocampus of rats that were wandering around in different kinds of mazes and boxes. And hippocampus is shown here. This first show a human brain, And you see hippocampus as a red structure, which is not on the surface of the brain. It is in the inner part of the cortex, so slightly underneath, but it's still cortex. And in the rodent brain, you see hippocampus as a sausage-like structure. It's also under the neocortex. So, but if you dig down here, you will see the hippocampus as this sausage-like area. And in that area, then O'Keefe recorded activity from single cells and displayed them on an oscilloscope and stored them in a computer, and then was able to find that these cells responded particularly and specifically to the location of the rat wherever it was in the environment. So I'll show you a movie to illustrate this concept. So what you will be seeing here is a rat. You see a box. A box is 1 by 1 meter large. In that box, you see it from above. In that box, there's a rat. You just see it barely now. The rat will be walking around chasing small chocolate pieces. And that's just a trick to, throwing out these chocolate pieces is a trick to have the rat wander around and visit almost every possible place in the environment. And then when this rat is walking around, then we'll be listening to the spikes or the action potentials of a single cell from the hippocampus. And each time the cell is active, fires an action potential, you hear a bop sound, and you'll also see a red dot appearing on the screen. So let's start the movie. So this is the activity of a single cell, and you can see that whenever the rat is in the upper left part of the box, the cell is active. It is not active when the rat is in other places. So in other words, this cell is active in one place in the environment, and that's why O'Keefe called them place cells. And he called this area of activity the place field of the cell. And this can be illustrated also by a color map or a rate map, as we call it, that shows in color the activity of the cell. So you can see that the red color is in the upper left corner and the rest is essentially blue. So this cell has a place field in one particular place, and then other hippocampal cells are active at other places. There are many thousands, hundreds of thousands of cells that have different preferences for different areas in the box. And that observation then led John O'Keefe, together with Linnell at the University of Arizona, to propose that these place cells are part of a Tolmanian map of the environment. So it's the building blocks for this spatial map that Tolman proposed. That was a major reorientation of thinking in the field, and it is still the fundament of how many people think about place cells. So, but a challenge was that these place cells in many ways stood alone. So no one knew how they came about because they were in many ways in the middle of the brain, far away from sensory inputs. So how do you create a place field? And one insight that started to help an understanding of that was the observation or discovery of head direction cells in the dorsal presubiculum, which is an area of cortex that is not so far away from the hippocampus. It's shown on the brain section here, so it's very near the entorhinal cortex and also has connections to the hippocampus. So in this area, Jeff Taube and Jim Rank discovered cells that fire in response to the head's, to the rat's orientation of its head. So whenever the rat's head was pointing in a certain direction, then the cell was firing. So you can see an example of this at the bottom to the left. This is a activity of one single cell. So on the x-axis, you see the orientation of the rat in 360 degrees around, and the firing rate of the cell. And you can see that this cell fires specifically when the rat's head is pointing about 270 degrees in the room, with reference to the surroundings, to the walls of the room, for example. So there is then, in other words, there's a directional coding and there's a place coding altogether. So how does all of this come together? Well, to try to understand this, we thought that recording activity in the medial entorhinal cortex might be an interesting way to go, because medial entorhinal cortex is the major cortical input to the place cells. So what you see here on this brain to the right, it's a rat brain. You see it from behind, and the colored area indicates the entorhinal cortex, and the medial entorhinal cortex is the upper half of this area. So we put our electrodes there and try to record just like in the same way that O'Keefe recorded the place cells in the hippocampus, and wonder then, what are the inputs to these cells? What would we find there? And of course, this area was also interesting because entorhinal cortex, medial entorhinal cortex, is an interface between the head direction cell system of the presubiculum and the place cell system of the hippocampus. It is in between. So perhaps it is possible to understand some of the coding and some of the integration of direction and place by recording in that area. And the entorhinal cortex in the human brain, you see it as the blue area very close to the hippocampus. So recording there, then that brings us to the year of 2005, when we observed that there are cells that have very, very peculiar firing patterns, grid cells. So you see an example of the firing pattern of a grid cell down to the right. So what you see is the trace in gray. You see the trace of a rat where it has been walking for a period of 30 minutes in a box that is 220 by 220 centimeters large. So during those 30 minutes, because it finds chocolate many different places, it covers essentially all locations of the box in all possible orientations. And what you can see is that, like the place cells, this cell is active at particular locations, but it's not just one or two locations as most place cells have. Here we have a number of locations, clusters of locations, and they are organized in a very, very special way, which it forms a hexagonal grid that covers all of the space. And I try to illustrate that by drawing these red lines on top of the grid cell firing pattern to the left here, where you can see that it's you can easily fit equilateral triangles on this, and the repeating unit across the environment is a hexagon. So it's a hexagonal pattern that repeats itself all over the available space for the animal. So this was, of course, not something we had expected, but it was very, very striking. It turned out that a very large proportion of the cells in the medial entorhinal cortex are grid cells, but they may have different phases, as we call. That means that they are shifted differently to each other. The peaks are at different locations in the box. They may have different spacings, that means, or frequency, that means that the distance between the peaks may be different for different cells. And they also may be oriented differently, so that the angle of the grid axis may be different from cell to cell. So this, of course, we found this very interesting and speculated that perhaps this could serve as some of the spatial inputs to the hippocampus. But it was also interesting because these cells obviously must be the result of some system that keeps track of the animal's movement, because how else can the cell fire exactly at the same spots when the rat is just walking around randomly and still have the cells fire at the same locations all the time? But it turned out soon that these cells are not alone. There's actually a number of specialized cells in this part of the brain. And already the year after, we found that although grid cells are predominant in the superficial layers of the medial entorhinal cortex, especially in layer 2, as we go down to layer 3 and layer 5 in particular, there are many, many cells that are directionally tuned. And this is the same type of cells that Taube and Jim Rank described. Those that fire only in certain directions, head directions. And you can see some of them here. So to the left, you just see where the recording was made in the brain. In the middle, you see the firing locations, and you can see there's not really any grid pattern. It's pretty scattered. And the important part of the plot here is to the right, where you see firing rate plotted as a function of orientation. So what you see here is for 2 consecutive recordings, you see that this cell in the top fires only when the rat has its head towards the left, and the same again on the second trial just below. And then the cell at the bottom here fires only when the rat is heading to the northwest or into the upper left, and again the same when it's doing that on the second trial. And there were also then cells that were both grid cells and head direction cells at the same time. I'm not going further into that. But these cells coexisted with yet another type of cells that respond specifically to the speed of the animal as it is running around in the environment. And if you focus on the left part here, you see the activity of 7 cells shown in different colors. And in the background for each trace, you see the speed of the rat as it's running around in the box in gray. So, and you can then see that the activity of these cells follows pretty much the speed of the animal. For example, focus on the yellow one, and you can see that it is almost a direct replication of the speed of the animal, which is shown in gray. So many of these neurons are interneurons, but they have really a striking linear relationship to the speed of the animal. So shown for each cell here, you can see that the color plots just show that they don't really have any preferred firing location, but the firing rate is linearly increasing as a function of the speed of the animal. And of course, having this speed information together with directional information might be important for keeping track of the animal's location second by second. But even those cells were not, that was not all, because there was yet another type of cell in this network that was completely different from the others, in the sense that there was no grid pattern, and they were not particularly directionally responsive or speed responsive, but they fired along specific walls of the recording box. So, or borders, any type of border that was placed in the environment elicited firing in those cells. So you can see an example in the upper left here. You see a cell that fires when the rat is along the right wall of the environment. And then if you stretch the environment, it's still along the right wall. If you stretch it the other way, it's still on the right wall. And at the bottom, you see the same cell after it's put in a new environment. Then it chooses the left wall. And if you insert a wall, as you see in the middle here, it even fires along that wall too. So these cells fire strictly along the walls or borders, local borders of the environment, and they are totally distinct from the grid cell. So you can never make a grid cell become a border cell or vice versa. But even those, that was still not enough, because in 2019, we found that there is yet another type of cell that doesn't respond to the borders specifically, but to discrete objects, salient objects that are placed in the environment. So these cells we call object vector cells, because they fire in a vector-like relation to these prominent landmarks. So here to the left, you see a mouse in a box, and there's a Lego tower here. And this Lego tower is also shown as a circle in the rate map to the right here in the color map. And to the left color, in the left part here, there is no Lego tower, and then it's introduced in the second trial here, and you can see that this cell becomes active once you put that Lego tower in, not firing at the Lego tower, but a certain distance to the left in this case. Other cells might fire to the right or on the downside or on the upper side, and the distance may also be variable. But what is peculiar to these cells is that the relationship, the vector relationship to the object is constant. So if you move the object, as you can see on the picture at the bottom, this object, the circle, is at different places in the environment, the activity field is still on the north side in both cases at about the same distance from the object. And if you plot the orientation and the distribution of orientations and the distribution of distances, you can then see here to the left that one row is one cell and color is the firing rate. And you can see that essentially the orientations are distributed across the whole range, and distances in this case was an almost 1 meter box was at least from 0 to 50 centimeters away from the object. So there are several ways of coding position. Grid cells are certainly one, but there are also other cells that code position by using a vector type of code instead. But that just brings me to what I will, which is the more interesting or more novel part of my talk. And the point I want to make is that throughout the last decade, we have been able to describe a number of elements of the neural map for space. So grid cells, place cells, border cells, speed cells, and so on. But what we really need to know, if we want to understand how the neural circuit works, is how do they interact? What is it that creates their firing patterns, and how do they work together to enable an animal to find its way? And then you can't do cell by cell. You can't study one cell at a time. You need to study populations. So before I go to the data, I want to say a few words about theory, because when you start recording activity from many cells, you need to be guided. Otherwise, I mean, there's so much to look for. Having a theory about how it all works is very, very helpful. And for the ideas, there are ideas about how localized activity is created. So I will begin with the head direction cells. Because these cells, there are actually models proposed already more than 20 years ago, 25 years ago, for how these cells might actually give rise to the directional signal in head direction cells. So these models are often referred to as continuous attractor network models, and I'll try to explain the concept very, very briefly. So imagine now that you have cells arranged in a ring, like you see here, different cells shown in different color. So they are arranged in a ring. This is not how they are arranged anatomically in the brain. You just put them in order of directional preference. So a cell that is tuned to the direction north, for example, is put here at the top. Then the model suggests that there is preferential connectivity between cells that have similar directional tuning, so that the red cell is connected strongly to others that are also red or orange, and weaker to yellow, and so on, so that similar cells excite each other. And this excitement of similar cells creates a bump of activity, because these cells keep each other active because they excite each other. And that bump of activity is kept under control, because in addition to this excitation, there's a wider inhibition that goes further than the red lines and stops it, constrains the activity. So here you have a way to create localized activity in a network. But if you then add incoming directional signals, angular directional signals, which there are in the brain. So you can imagine a clockwise signal that could come in, a clockwise speed signal or a counterclockwise speed signal. The idea is then that this signal would move the bump around in the ring. And this is still, of 25 years later after this was proposed, and proposed among several people, but Bill Skaggs, Bruce McNaughton in Tucson were among those who proposed how this could work based on earlier work for similar models in other systems. So this is how you could create a directional signal. And in fact, there is some evidence for direct observation of such ring attractors now in flies. I will not tell more about that, but use this as a background for introducing two-dimensional models for localized activity. So the idea is the same. Now you arrange the cells according to where they fire in a box. So all the dots here are different cells, and they're not arranged in this way in the brain, but here they are plotted in the way they are, where their firing preference is in the box. And again, the core idea is that there is strongest connectivity between those cells that code for the same location or similar location, and then it tapers off as you get away. And again, you have a ring of inhibition around here, not shown, which then gives rise to the idea of Mexican hat, because you have strong excitation in the middle here, and then you have inhibition or this circular valley around. So this is a way to create localized activity in a network. And again, if you have directional signals and speed signals coming in, you then have a way to move this bump of activity in the around. So this idea was then introduced by Bruce McNaughton and others in the McNaughton and Barnes lab as to account for localized activity in place cells in the mid-1990s. Then there were some challenges with that model for place cells, and one of the challenges is that there are different place cell patterns for almost every environment that a rat encounters. So there would have to be many such networks. So that would be possible, but it would require quite some capacity. So it was then interesting, we thought, then when we found that there are actually place-like patterns in the entorhinal cortex, but that these have a grid-like pattern. And I still remember that Bruce and Carol and Maybrit and I discussed this very intensely when they were visiting sometime in 2005, 4 or 5. So, and the idea here is you can just extend the model for the place cells by suggesting that either these bumps of activity can arise independently in different areas of the network, and they will keep each other apart by this ring of inhibition around, and it will naturally form a hexagonal pattern. Or alternatively, you may suggest that actually there is only one bump, but the network has a circular shape, so that the right end of the network just connects again with the left end, and the top end just connects again with the bottom. And if you sort of fold it around, you can get a toroidal or a torus pattern that I will come back to very soon. So, but let us now go to the data. So in order to test such models for how cells work together to create direction or place signals, we need to have access to the activity of many, many cells at the same time. And that was actually the reason why Maybrit and I came to Tucson a long time ago in 2001, because the Tucson lab of Carol and Bruce was pioneering research on recording from many, many cells at the same time in the hippocampus. So this shows a recording from one of their papers, where I think there were 80 cells recorded, which was amazing, we thought, and I don't think Maybrit and I ever obtained that when we were in Tucson. but that was the gold standard at the time. However, even 80 might be too little to understand such attractor dynamics. So now we are participating in a consortium that develops a new type of probe for recording activity in the brain, referred to as Neuropixels probes. And these probes already have allowed us to record more than 1,000 neurons at the same time. So it's a massive extension of how much data you can collect from the network. So these are probes with multiple sites of recording, 384 sites on each probe, and you can have at least 4 shanks of probes into the brain at the same time next to each other. So what you see to the right here are rate maps similar to the one that you saw next to the movie that shows in color the activity of the cell, but each square is a rate map for one cell, and in total here you have 1,064 cells at the same time. So then what can you do when you have so many cells at the same time? Well, you can then go back and test what is the organization of the activity among all these grid cells, if you happen to have many of them in a recording. Are they organized in a toroidal pattern? Do they map onto a torus pattern of activity, or is there some other topology underlying it? And for that, we collaborate. First of all, it's Richard Gardner, who is a postdoc with us, who is doing the recordings and pioneering this work. Then it is 3 people from the maths department, and then Yoram Burak from the Hebrew University of Jerusalem. And together, they have started out now trying to analyze what is the structure or activity underlying the grid cells. So using Neuropixels data from, in this case, 2 rats with respectively 1,075 and 1,306 cells, recording from several modules of grid cells that have a similar spacing among them. So at the bottom left, you see a raster plot that shows in each line is the activity of one cell. And this might, for example, be the one with 125 cells. And then on the x-axis, you have time, and you can then chop through this or define time cycles of, let's say, 10 milliseconds, and then look at the combined activity in all these 10 milliseconds, and then plot it. Well, let's wait a little bit with that one. So if you can plot the activity of all those 125 cells at the same time, and in principle, those the activity of those cells can be combined in an endless number of ways. So it might be truly high dimensional. And if you plot the activity in a 125 dimensional space, it actually is almost possible to work with. However, it turns out that there are just certain patterns of activity or combinations of activity among those cells that repeat itself all the time. And so if you want to see how can we explain this variance, you can actually then perform some so-called dimensionality reduction analysis, where we break down the activity to the core dimensions that explain most of the variance. So using a principal component analysis, and in this case, we are doing it in 2 steps and finding the key dimensions, and then doing another dimensionality reduction called the UMAP. And if you plot the 3 most important components, then what you get here is actually something that does look like a torus. And as an example, the red and the green times are just shown as points in this point cloud. However, plotting it that way doesn't lend itself easily to quantification. So in order to quantify the activity here, then we are using something called persistent homology. So it's a mathematical approach to identify holes in a point cloud. So the idea is that you plot the point cloud in n dimensions. In this case, I just show 2 dimensions. And then you draw balls or circles around, and then you expand the circles, and then you try to you look for holes between identified circles. So now you see there's a hole in up here in the upper left part, and you can now say a hole has been started, and you plot that here with the bar here. A hole has started, and now it stops, it's filled. So then, but then you can continue like this, and now these lines, these are all connected, and you start to get another hole, and then you continue this process again and move on, and until it's all filled, which happens very soon. There. Oops, sorry. And what you get there, and the idea is that sorry, let's see once again here. So the length of these bars illustrates how big the holes are in this point cloud. And why do this? Well, the fact is that with this approach, you can actually predict what kinds of holes you would encounter if this was a torus. So torus is defined by two types of holes. So in one dimension, you have two circles, they're orthogonal to each other. And in 2 dimensions, you have this donut-shaped structure here. So you can then look at these bars that we defined and then see where do you get a long bar. And it then turns out for all these data sets, this is data sets from Animal Roger, module 1, module 2, module 3, and then another rat, module 1. And then you can see that you have two very long bars in one dimension and one very long bar in the second dimension. And that is exactly what you would predict to see if this was a torus. So you just have to take my word for this, but this is topology. And note now that you see the same actually in all of the data. So two rings in one dimension and one torus in the 2 dimensions. So and if you now test this not only when the rat is walking around in the environment actually, but even in other circumstances when there is no grid pattern, such as when the rat is sleeping or when he's walking in a circular maze and you can't easily see the grid pattern, you can actually see that it's the same thing again. It's 2 long lines in the first dimension and 1 long line on the second dimension, and you can see how this is preserved during slow wave sleep and also during REM sleep. So this suggests that there are the holes of this point cloud actually correspond exactly to what you would expect if it had a toroidal shape, and this toroidal shape, the activity structure is maintained even if the rat is not walking around any longer, it's just sleeping or doing whatever, it is always there. And knowing that, you can then proceed to actually plotting the activity of the cells onto the torus. You can infer the torus mathematically, and then you can plot the position of firing for each cell onto that torus. So that's what's done here. So it's shown as a rhombus, but this is just an unfolded torus. You just wrap it up in the 2 directions. And then you can see that the activity of each cell is at that location. And then the same when it's walking in a circular maze. And if it is sleeping, you can still see that the relative locations here are very much the same. You see that also here to the right. It's very, very similar activity. So the activity of this network on the inferred torus is the same, whether the animal is walking in a box, whether it's walking on a circular track, or when it's sleeping, slow wave sleep, or REM sleep, all the time. And that suggests then that the grid cells are actually mapping, the activity is mapped onto a low-dimensional manifold that can be described as a torus, and that activity structure is maintained regardless of the sensory inputs that come in. So this is very much in agreement with the predictions from continuous attractor models. So then let me switch gears. I want to say now, let me see how many minutes are left. 5-10 minutes, would that work? I will at least try I'll try to say a few words about the hippocampus. So the hippocampus is very different from the medial entorhinal cortex. I will not, in the interest of time, I will not show the data for it, but just tell you that while grid cells map onto this low-dimensional manifold, place cells are very different. There are different maps of place cells for almost every possible environment that the animal is in. So then, given that, that is very actually consistent with what we have known about the hippocampus for a long time, since the mid-1950s, when patient HM received a surgery into a hippocampus, and hippocampus was removed in order to rescue patient HM from serious epilepsy. So this led to a severe loss of episodic memory. And now that we know, the hippocampus is actually a structure that receives all this spatial input from grid cells, from object vector cells, from border cells, and all that. And space is a very significant fundament of episodic memories. So that is fine. But there is one other element that is missing that I haven't said much about, namely time. When you have a memory of an experience, then there's not only space in it, it's also time. And so that brought us to look further into the representation of time. So there are studies that have shown that time is also expressed in activity of hippocampal cells, hippocampal place cells. So we went up into the entorhinal cortices and actually focused on the lateral part of the entorhinal cortex, because that area has not been well investigated. We know very little about it. But we had a student, Edvard, who recorded activity there for a long time while rats were walking in boxes, in a series of boxes over a period of approximately 2 hours. And then he recorded activity from those cells in these long recordings, and found that some of the cells actually had very strongly temporally patterned activity. So activity went up and down each time the rats switched from, each time the rat was in the black box or in white box or in the black box, you can see that it's ramping up all the time, as if it is monitoring the time that has passed. And other cells had other patterns of activity that could also be mapped onto time. And if we did an analysis, GLM, a general linear model, try to break down the significance of different types of input to these cells, it actually showed that while position, of course, was very strongly involved in explaining the activity of cells in the medial entorhinal cortex, where the grid cells are, and in the hippocampus, CA3, then time does not account much for activity in hippocampus and medial entorhinal cortex, but does explain a lot of the activity in the lateral entorhinal cortex. And that then also can be seen if we decode from the activity of lateral entorhinal cells, decode the activity and guess which time is a recording being made based on the activity that is the combined activity of cells that we have in the recording. So we can use a machine learning approach where we take activity from many time bins of activity, train the network, and then ask the network for a new recording. When is this being taken? And then for the lateral entorhinal cortex, it is possible, based on the combined activity, to guess almost in all cases pretty accurately the time of recording during this long series of data acquisition, whereas this does not happen so well for the hippocampus and medial entorhinal cortex. So let me skip next slide. But then finally, just say a few words about the temporal organization of activity in the entorhinal cortex and also in the medial and the lateral, because the data, actually activity in this network is strongly organized into sequences. And some of these sequences are so striking that I wanted to share one of those with you in the end. And this is especially the work of Flavio Donato and Soledad Gonzalo Cogno, who is a postdoc in our lab. Flavio was there before. What they did was that they recorded from mice while they were running on a ball, head fixed, and at the same time they then used two-photon microscope and the prism approach to image the activity of hundreds of cells, many hundreds of cells at the same time while the mouse was running on the ball. And the mouse was running in complete darkness, nothing to see, no goals, no food reward, nothing, just spontaneous running and occasional breaks in between. And then recording that activity, you see the cells blinking here, it's fluorescent calcium. So it's monitoring GCamp6 activity of these cells while the mouse is running. And this is the last data slide, so I'll just show you now. First, a raster plot which shows the activity of more than 400 cells as a function of time. So time is on the x-axis, and the neuron number is on the y-axis. And you can see that's not very much pattern in this. It is just looks like a random mess of activity. But then again, using dimensionality reduction analysis, or in this case, principal component analysis, or can also use other types of analysis, then what they showed was that if you identify the principal components, you then break the activity down into time frames, which are the vertical, the columns here, and then in the rows you have the cells, and then you break this down into a few dimensions that explain most of the variance, and then in this case just focus on the two most important dimensions, and then define the angle between those two dimensions, and then sort the cells according to how strongly they load onto either one of these components or the angle. So here is the angle because that gives the clearest results of all. So the one that has the strongest loading onto the angle is then given one number, and then they are arranged according to how strongly or what angle they actually map onto in this data. And what we find then is this. So you can see that if you define, if you rearrange the cells according to how they map onto these first two dimensions, the angle between the first two dimensions, you can actually see that there is a pattern or activity that repeats itself over and over and over again all the time while the mouse is running. So with a cyclus of approximately 40 seconds in this case, so it's a very slow waves of activity that go through the network. And actually this is circular, so this is just the same data shown with time shown in color. So you have time on this axis, and then you have the two dimensions on the two other axes. So this shows then, in other words, that this network of the medial entorhinal cortex, and also probably lateral, when the mouse is just running with no stimulation, complete darkness, and even when it stops, actually, there are these cycles of activity with a period of often around 30, 40, 50 seconds that repeat themselves all the time. So that suggests to us then that while there may be attractors that may underlie head direction cells and grid cells and so on, there are perhaps also cycles of activity that are much longer and may perhaps reflect networks that we don't know about yet. So these do not correspond to any anatomically defined networks that we are aware of, but obviously suggest that the activity of the entorhinal cortex is strongly organized in time also. And that is then is still ongoing work. We are wondering what this means. But let me then just round it all off by saying that we have now the tools for exploring the elements of or exploring how many cells work together to create a sense of space and a sense of time. When I say time, it is actually the organization of experiences in sequences probably. But whatsoever, these are inputs to memories in the hippocampus. So without those, if you don't have space, if you don't have time, if you don't have memories, that at least would at the very least lead to an altered state of consciousness. So that's what brings me back to the topic of this conference. And let me then just finalize by thanking all the people who have been involved in this work, and Moser, and then a number of other people. Let me then remind again about the very powerful collaboration we have had through the years with the Tusen Lab, with Carl Barnes and with Bruce McNaughton, and also others I have mentioned through the talk. So with that, thank you very much for the attention, and I think I will stop share now.
Thanks, Edvard.
Glad to see you're still there. I was wondering if I was just sitting talking to myself.
No, we were watching it. Stuart was here too. Okay, we have a number of questions for you that have come up, and are you willing to take a shot?
Sure. It's the Q&A. Now I open that. Yes. Okay.
Okay.
All right.
So the first one I'm going to read to you. The hexagonal patterns of grid cells change scale in different layers of the entorhinal cortex. Is that correct?
Could you say it again?
The hexagonal patterns of grid cells change scale in different layers of the entorhinal cortex.
In different anatomical layers. No, I think they are actually strikingly consistent, so that we did address this in a study that was published almost 10 years ago, where already then there were enough cells, both from at least layer 3 and layer 2, and it's remarkably how consistent they were. So I think they are so strongly linked, the layers, that they operate in many ways as one unit. But that said, the grid cells are organized into modules of cells that have a similar spacing and also other similar properties, and they are intermixed, so that in any given location you may have cells that maybe have a large spacing, and you may have cells that have a slightly smaller location, because they are not completely separated from each other. And this is also reflected across the layers.
Okay, so the continuation of the question is that some think that there are hexagonal patterns representing information in microtubules inside neurons or nervous cells. Could these be a smaller scale, high resolution of grid cell maps inside neurons?
Well, I would just say that there are hexagonal patterns all over nature, and I think I mean it's a natural. It's a kind of equilibrium state that ends up for many different reasons. So I think I wouldn't make an obvious link between those two, or at least I couldn't think of a mechanism how they were linked. But I think it is interesting to see how many phenomena in the world, in the brain or outside the brain, that actually end up with a hexagonal pattern.
And so the corollary of this question is, do single organisms know where they are in space, like microtubule hexagons?
Well, I don't know what a microtubule hexagon knows. That's very hard to say. I think the grid cells are part of a system that allows us to know where we are in space, but it is just part of it, and actually knowing where you are involves very large parts of the brain that have to work together, but it's an important element.
So I have another question for you from somebody that I don't have their names, but the person says, I have a question about location mapping in entorhinal grid cells. Do you think they can serve a physical basis for understanding what selfhood is? And how would that account for the self as an observer to whom the location model is presented?
I think if selfhood is a feeling of how you are different from others or different from the world, then I would say that these cells, any spatial cell, but also the grid cells, likely are part of it, because without a sense of location, I don't think, and also without a sense of time, I think it's very hard to imagine that you can perceive yourself as distinct from the rest of the world, and especially knowing that space and time are so important elements of episodic memories. So if episodic memory then should also not work, then it's very hard to imagine what is left.
Right. Okay, next question is, how do you determine that the place cells fire in response to a location or position rather than in response to architectural space? That is to say, in response to processing architectural spatial dimension.
I'm not sure I understand what architectural space is. That you have to explain to me.
Okay, I can't because I'm not the but maybe the questioner can type that in a box below and help us out with that question. I'll go on to the next one. Is there a correlation to the vector technologies, mathematics, and I don't know, I can't read this, geometries used in 3D gaming environments? So are we mimicking the activities in the brain there? And is that why VR is so immersive and disorienting? So is there a correlation between these vector technologies and mathematics that they use in gaming?
Well, I don't know much about that technology, so I think I'm the wrong one to be asked about that. But I think it is true that you can trick the brain actually to believe, if you provide enough inputs that are similar to the real world, the brain will fill in the rest and actually make you believe that you are moving in an environment, even if there are other inputs that don't really correspond to the real experience.
Okay. So I don't know the abbreviations here, but it says, can CAN models describe WN? Do you understand that question? Or is working memory similar to a rat navigating a circular maze? Precession angles and 2 pi.
Well, that sounded very complicated to me. So this, I think I need to have that repeated. So CAN, that's a continuous attractor network models, whether they are similar to
WM, is that?
Oh, is that working memory or?
I'm not sure.
No.
I need more feedback from the questioners. Are we getting more questions?
Yep, there's one.
Okay. So if those who ask the questions can follow up and we can re-ask, but here's another question.
Mm-hmm.
A few questions related to studying the topology of network. 1, what would you expect from distorting the room in real time with visual or movement related illusions and or a morphing room? And 2, similarly, have you considered administering various drugs? Most would be interesting, but particularly the psychedelics, which have phenomenological warping of space and time.
Yeah, no, that's an interesting question. I guess if I never tried these drugs myself, but I assume that you can see walls just coming in and disappearing and changing shape and the whole thing. So how that would be experienced by a rat or by a human subject is interesting. But I think based on the data that we have, I'm not sure I would actually think that in response to such experiences that the grid, that the topology would change, because the topology, the torus topology, for example, or the ring topology is a result of how the network is wired together. So if you wire the cells together in a special way, in the way that I described, then you get a certain type of activity. So I think whatever external inputs you give to the system, you can't escape that. So I'm pretty confident that you will retain that topology whatsoever.
Mm-hmm. And for the first part of the question, though, you have changed rooms and you have shrunken rooms and so forth, but have you done that while the animal's sitting there? Or?
Well, that's actually a good point, because I think whether the animal is sitting there is the critical difference. Because we are doing many, and Carol also, everyone in the field does that type of experiments where you try to trick the rat and change the environment, and it comes back and it's confused, and then you use that confusion as a way to find out how the system works. But if the rat is sitting there while you're compressing the environment, then the rat will probably figure out what's going on, and it will calibrate for that, so that you might get different results of, depending on whether the rat is there or is not there.
Yeah. I have another question here. Does your research support the idea that perception of space has a multidimensional nature?
Perception of space, I would say, I'm not sure whether the perception part is important, but I think the data that I showed illustrates that in many ways this system, the entorhinal system at least, is strongly low dimensional. I mean, it's striking how similar the structure of activity is regardless of the experiences. So, but of course, that doesn't exclude that there are other dimensions that come less through. Maybe they are not, do not account for so much of the variance, and we lose them when we use these techniques to reduce the number of dimensions. That's very well possible, but I think it can be compared with the other structure that I just very briefly mentioned, namely the hippocampus, where the place cells were discovered. Because here, what is so striking is how many combinations of activity you can actually have. Each time you take a rat to a new room, you have a new combination of activity. So for the hippocampus, the expression of activity is very high dimensional. You can combine it in so many different ways, and that is space in a very different ways. And it's a kind of spatial coding that is very suitable for memories, because you can store so many different patterns in such a system, whereas the rigid entorhinal system is useful for encoding location, but I mean, its strictness or even lack of plasticity in some sense is perhaps not where you would put all your memories. But then if you then ask how is this perceived, the perception of space, I'm not exactly sure what you would mean by that. Perception to me is just how the rest of the network reads out the activity. So in that sense, perhaps it's not different from what I said.
Mm-hmm. So thank you for this really beautiful lecture, Edvard, and your new data are absolutely spectacular. I wish we had more time to talk about it, and I wish there was a neuroscience this year. Edvard always gives me a private tour of his posters at neuroscience, and I get the latest stuff there. But I'm glad.
I had hoped so much to present this at a real talk in Tucson, but well, hopefully that day will come soon.
2 years from now.
April 18th.
Put it on your calendar. April 18th, we'll have you back. Okay, thanks so much.
Thank you, Edvard.
Okay, all right. Bye-bye.
Bye-bye.
Okay. Nice to see you all. Nice to see you, Cal.